Hello students, how are you all? Good morning guys. How are you guys? Good morning all. How is your preparation guys? How is your preparation? Okay. So before going to start the class, I have told you in the last class that from the principle of inheritance and the molecular basis of the inheritance, you are going to get 30 to 40 marks. Okay guys. So be prepared well in this topic. Okay. Fine guys. So today also we are going to discuss about the very important topics on these chapters. Let us study. Okay. Fine. So before going to start the class, can you tell me whether I'm audible and screen is visible guys? Okay. So tell me whether I'm audible and screen is visible. You two people, tell me. And how is your preparation going on, guys? How is your preparation, Nandish? What about others in the YouTube channel? Okay, guys. So let us start the class. In the last class, what are the things we have studied? In the last class, we have studied about the Mendelian, right? Mendelian inheritance, like the 3 laws we have studied, right? Can anyone tell me which are those 3 laws? Can anyone tell me, guys, which are those 3 laws? Yeah, very good. First law is the law of dominance. Second law is the law of segregation. Third law is the law of independent assortment. In that we have studied that one character will assort independently when it compared to the other character. Right guys? When two characters are there, so both the characters will assort independently. So why also have told you, right? Because both the characters on the chromosome are in the long distance, right guys? When they are in the long distance, they will inherit it independently. That thing we have studied in the last class, right guys? Yes. So then let us go to the non-Mendelian inheritance. Remember guys, some of them will confuse about the non-Mendelian inheritance. If you get any confusion in between, kindly ask me. Okay, fine. So Nandish, we are directly going to the topic part, not to the questions. Fine. So let us go to the non-Mendelian inheritance. So there are how many types of the non-Mendelian inheritance are there, guys? One is incomplete dominance. Second one is co-dominance. Third one is multiple allelism. Fourth one is polygenic inheritance. And fifth one is pleiotropy. Okay, fine. So whenever there is a difference between the Mendelian inheritance and the non-Mendelian inheritance, you have to remember that in the Mendelian inheritance, the phenotypic ratio will be different and genotypic ratio will be different, right? So when it is comes to the non-Mendelian inheritance, where the phenotypic ratio and the genotypic ratio will be same, okay? And so here we are going to get the intermediate progeny, okay guys? We are going to get the intermediate. When it is compared to the Mendelian inheritance, there is no intermediate are there. Remember these two things, very important. Fine, let us go one by one, okay? So let us first go to the law of dominance. When we are talking about the type of dominance, there are the 3 types, guys. Okay. One is complete dominance. So that we have studied in the case of the Mendelian inheritance, right? So when capital T, small t is there. So in the F1 generation, heterozygote. So that is the capital T and the small t is there. So where the capital T is dominant over small t. Right or not? Tell me right or not? Yes, right. So here what happening in the complete dominance, so that is a Mendelian inheritance. Coming to the incomplete dominance and the codominance, those two are the non-Mendelian inheritance. Let us study one by one together. Okay, fine. So let us come to the incomplete dominance. So guys, what do you mean by incomplete dominance? If you know, can anyone tell me? Just give an example, guys. What do you mean by what is mean by incomplete dominance? Can you tell me, guys? No dominant, no recessive. Very good. So what happened here, guys? Not like that we have a capital A. For example, you take capital R. Okay. Capital R is there. Capital R is there. Right. So this will give the red color. Right, guys. And you know that small r is there, it will give a white color, right? So what we have studied in the Mendel inheritance, if it is a capital R, small r is there, so during the law of dominance, what will happen? So only the capital R will express. So then we all get the progeny that is a red color in the F1 generation, right or not? In the F1 generation, we will get the red color when it is compared to the Mendel inheritance. But what happening in the incomplete dominance? Here, where dominant allele will not completely repress the recessive allele. Remember, dominant allele cannot completely dominant over the recessive allele. Okay, in that case, what will happen? Some small amount of the recessive allele will also express, some small amount of the capital allele also express. So here we are going to get the, in the F1 generation, capital or small r, we are going to get the pink color flower, right guys? So here what happening, where dominant allele completely not repressing the recessive allele. Remember guys, okay? So which are the example for the incomplete dominance? In the exam, they may ask you like this, which are the example for the incomplete dominance? Remember guys, snapdragon, we also call it as a dog flower, okay? And mirabilis jalapa, that is 4 o'clock plant. These are example for incomplete dominance. Okay now. So there. So where dominant allele is not completely repressing. That is the thing. Okay. So here what is happening? I have told you the cross between the red color and the white color flower. Where in the F1 generation it will give the pink color flower. And what is the genotypic ratio and what is the phenotypic ratio? Before only I have told you that both are same. Right guys? Both are same. That is phenotypic ratio is 1:2, 2:1. One is a red color, two is a pink color, and one is a white color. So genotypic ratio will be a same 1:2, 2:1. Okay guys. So what here they are telling? Where capital R not completely dominant on the smaller. Okay guys. In P plant also we can see this type of the incomplete dominance. How many of you understood this incomplete dominance? Give me a thumbs up in the YouTube channel also, guys. How many of you understood incomplete dominance? Okay. So, give me some thumbs up or something, guys. Then only we can go fast. Okay. Fine. So, let us go to the next one. That is the co-dominance. Co means what? We can call it as a colleague. Colleague means what, guys? Where the two persons are working in together, we will call it as a colleague. Right? Colleague. We will call it as a co-worker. Right? Co-worker. So what happening in the co-dominance? Both of the gene. Here there is no recessive allele or there is no dominance allele. Here what happening? Both of the allele will express together. So it is like you also same, I am also same. Both will be a dominant now. It is like a both two are friends. Okay. So when sister and brother is there, sister is the dominant there, right? So when both are the friends are there means what will happen? Both are dominant to each other, right or not, guys? Same thing happen here. The example is the ABO blood group. ABO blood group. What will happen here? You can see here. So it is the inheritance of the both allele. Both allele. Okay. So here you can see in a AB blood group. For example, if you take AB blood group, AB blood group. Okay. So IA is there and IB is there. What we will call it as? AB blood group. AB blood group. Yeah, another example I will take. Wait. AB blood group. Okay. So IA and IB both are going to express together. Okay. So I think you know this example, right guys? So because what will happen? So for you know that where the this blood group is mainly due to the glycoprotein which is present on the plasma membrane of RBC, red blood cell, right? If it is producing the one type of the glycoprotein, it will give the A blood group. If it is producing the another type, it will give the B group, right? When both are giving, both the glycoproteins are giving, then we will tell as AB blood group, right guys? Okay, so let us study about that. This is the same thing I have told you. Where O type of the blood group will not produce any type of the glycoprotein. So that's why we are calling as the O blood group, right guys? Okay. So here in the example, you can also observe here where father is having the A blood group. So here the father may give IA or I, right guys? So this is IO, right guys? IA and IO. This will give rise to the blood group. So that is the A blood group only, right? If it is IA, then also the blood group will be A blood group only. Right, guys? IB, IO, then also give rise to the B only. IB, IB, then also give rise to the B blood group only. Right. So this is what we will see in the codominance. Let us go to the another example. Have you seen the coat color in the cow? Have you seen the coat color in the cow, guys? That we will call it as a variegation. So there will be two types of the coat colors are present, where you can see the black and white color mix. Have you seen or not? Give me a thumbs up if you've seen means. You two people, give me a thumbs up. Have you seen this means? Black color. So you can see the cow is black color. Okay, cow is black color. And you can see white patches on the cow. How many of you observed that? Tell me guys, how many of you observed that? Black color cow on that white patches, or white color cow on that black patches. How many of you observed? Tell me fast. Nandish, have you observed that? Yes, Nagveni observed that. Very good, Nagveni. So what about others, guys? Nandish. What about others in the YouTube, guys? That is due to the codominance. What happening here? The color which is giving to the black color, that is also dominating. Whereas the color, white color which is giving, that is also dominating. Both of them are expressed together. We can't differentiate that whether it is having the dominant only, right? So here black also dominant, B also dominating, right? So we will call it as co-dominating, right guys? So black color also we will get, and the white color also we will get. So in the flower also you can take that is having the red color flower crossed with the white color flower. In the obtaining the progeny we are getting, that flower is having the variegation type. We will call it as a variegation type. Means, so there is a mosaic-like appearance. Okay. So that mosaic-like appearance is due to the codominance. Okay, guys. Mosaic-like appearance is due to the codominance. Always remember. I do. Yeah, Nandish also seen that. Remember, guys? So if it is giving the different color, that is the incomplete dominance. Okay. So when it is giving the blending of the, sorry, not blending. If it is comes to the blending of the two color, that is incomplete dominance. When they are asking about the mosaic-like appearance, you have to tell that codominance. Do you understood this, guys? Do you understood this? How many of you understood? Give me thumbs up, guys. If you understood this means codominance. What about you two people? No one is answering in the chat box. What happened to you guys? Yes, Nandish. Mosaic means what? Mixed. Here you can see. Here I have given the diagram, right? That is a cartoon diagram. See here. Father is the blue color, and another, I'm sorry, one parent is blue color, and another parent is white color. So progeny obtained, that is having the mixed type, right? So mixed type it is having blue color also and white color also. That we will call it as a codominance or the variegation, mixed type or the mosaic type. Do you understood this, guys? Do you understood or not, guys? Fine. So always remember, if they are asking blending of the character, incomplete dominance, mosaic, codominance. Yeah, fine. So let us go to the multiple allelism. What do you mean by multiple allelism? See, guys, one gene is having more than two alleles. We know that. For example, one gene, if I take the one gene, 1 minute, black only coming. Yes. So if it is one gene is there, okay. So we have studied in the height, right, guys? So one gene is dominant. So here it is having the two alleles, right? One is capital A and another one is a small a, right, guys? So there are two alleles, capital A. Either it is having the capital A, capital A, capital A, capital A, or heterozygous it is having the capital A, small a, or another one is having the small a and small a. Two alleles are there, right? But what happened in the multiple allelism? It is having, the gene is having more than two allele, okay? So observe here, for example, ABO blood group. For example, if you take AB positive, how many alleles it should have? Tell me, guys. AB positive. Tell me, guys. AB positive means how many blood group, how many things it should be have? See here, it should have the A, right? It should have the A allele, it should have the B allele, and it should have the O allele. Right or not? Right or not? Is polyallele is differ from the multiple allele? Yes, yes, yes. Polyallele and multiple allele is same pa. Whereas the polygenic is different. Polyallele and multiple allele is same only. Polygenic is difference. Okay, fine. See here, guys. If you take the AB positive blood group, in that what will happen? A allele is there, IA is there, IB is there, whereas the IO is there, right? For single character, whereas for the single blood group, it is having 3 allele, right, guys? 3 alleles are there. So that's why the example is ABO blood group. How many of you understood? How many of you understood, guys? From the YouTube, answer me. How many of you understood? Yes, Lakan. Yes, Lakan. Yeah, that in the codominance, AB blood group will come. AB blood group, where IA and IB, both of them is codominating. Codominating. If you take ABO blood group, then the example is a multiple allelism. AB blood group will comes under the codominance also. When you are talking about the ABO blood group, it will comes under the multiple allelism. That only I am giving the example. If you take AB positive blood group, so there are the 3 alleles are there. I allele are there, IB allele are there, and IO allele are there, right? So that is what I am telling Lakan. When you take only AB blood group, so then you can see the codominance. Have you understood, Lakan? Okay. So that only they are telling. See here. In the ABO blood group. Guys, always remember, if they are asking only AB blood group, codominance. If they are asking ABO blood group, multiple allelism. Okay, fine. In an individual, 2 alleles are present, but in the multiple allelism, more than 2 alleles are present in the population. Okay, guys, fine. So, and this also you can tell that. For if you take the B blood group only, so it is under so many alleles, right? Either it can be a IB, or it can be a IO. That is also the multiple allelism only. Fine. So yes, do you understood, Lakon? Fine. So now let us go to the polygenic inheritance. What they are telling, we all know that, right? One protein, for example, we know that one character is monitored by the one protein, right, guys? Yes or no? Yes or no? What is HH? Height, okay, sorry, I have taken the A, sorry, yes, capital H and the small h, guys, yes. Nandish, now you are saying, fine, no problem. Here, you can take the capital H and the small h, guys, sorry, fine. So let us go to the polygenic inheritance. What happening here, guys? For one character, more than one gene is responsible, okay? More than one gene is responsible. So those type of the inheritance, we will call it as the polygenic inheritance. In the polygenic inheritance, the example are the human height, human skin color, and the hair color. Hair color will also come. Human skin color will also come, and also the human height also come. Now there is the work for you. Now you tell me, if you take the height of the person, can we see that only the 5 feet person is there and the 3 feet person is there? Only the 2 types of the height persons are there, guys. Tell me, only 5 feet and only the 3 feet. Can we observe that only 3 types of the height are there? No, right. In the diagram only you can see that from the very height, having the very height person to the short person is there, right, guys? So here the intermediate height we can see, right, guys? In the color also you can see that some people are very dark, right? So we will call it as Negroes, and some people are wheatish color, like in the India we can see the wheatish color people, and in America and all the thing we can see the albino, right, guys? Turkish, we will call it as a Turkish also. So that is albino color, very white in white skin color, right? So there is, you can see the gradient you can see, right, guys? From the white to the, from the small little white, then to the whitish color, sorry, wheatish color. So then go to the little black, then go to the then go to the Negro. So that is very black, right guys? So their gradient is present, like the intermediates are present, right? So then how it is possible? We can't see only the Negroes and we can't see only the albino people, right? So this is due to the polygenic inheritance. Let us study about that. The examples are human skin color, human height, and the head color. Okay, fine. So here the skin color is maintained by were given by the three genes, guys. One is capital A, capital B, and the capital C. Okay. So if the peoples are having capital A, capital A, capital B, capital B, capital C, capital C. So they are very dark in color. Okay, guys. So they are very dark in color. Okay. So then coming to the small a, small a, small b, small b, small c, they are very, we can call it as very light in color. Okay, very light in color. So that is albino. Okay, fine. So let us go to here. So let us see here. What about the wheatish ma'am? Capital A, small a, capital B, small b, capital C, small c. So these are having the wheatish color. We are having the wheatish color, right, guys? Are you agree? We are having the wheatish color for the color of the skin, ma'am. Yes. So there, when we are talking about the genotype, that is correct. So those are different parts. Usually it is ABC only. Okay, Nandesh. Fine. So let us go to the next. Whatever there in the your textbook, that you can study pa, because in the NCERT basis only they will do the correction. Okay, that only I have told you now. So in all the, if all the dominants are there means it will give the dark color. If the all the recessive one are there, they will give the light color. And in between the heterozygotes are there means they will give the wheatish color. So see here, that only I have told you, same thing. So here you can see how the graph is going from the albino to the very black color. Okay. So you know guys that wheatish color we also call it as mulatto. Wheatish color we also call it as mulatto. Remember. Okay. So this is the crossing over they have did with the mulatto. Both are mulatto only. Okay. So in that we can see the 7 type of the phenotype and the 27 type of the genotypes. Okay. So here you can see that. Fine. So this is about the polygenic inheritance. How many of you understood what is a polygenic inheritance? Give me a thumbs up, guys. How many of you understood what is polygenic inheritance? Yes, Nadesh. Godhi Varna Sadharana Maikatti. Yes. How many of you understood, guys, from the YouTube people? Give me a thumbs up, guys, if you understood in the chat box. Fine. So let us go to the pleiotropy. Nagveni, do you understood what do you mean by polygenic inheritance? Fine. So let us go to the 1 minute. Yes, fine, ma. Thank you. So let us go to the pleiotropy. Pleiotropy means what? You know. So I'm the one person. I will take care of everything. Okay. So it's like I'm the cooking. So I will work as a cook also, and I will teach also. I will guide you also, and yeah, I will behave like a friend also. Okay. So if this all the work is done by me, okay. So then my work we will call it as a pleiotropic, okay. Whereas one gene controlling more than one character, okay guys, where one gene is responsible for more than one character, we will call it as a pleiotropy. What does this mean by pleiotropy, ma'am? What are the examples? Let us study here, okay. So see here, where single gene exhibit multiple phenotype. So those expression we will call it as pleiotropy, okay. So you know guys, the gene which is exhibiting the pleiotropy, it is usually involved in the metabolism. It may be an enzyme, okay. It usually may be a enzyme, okay. Protein, you know that enzymes are the protein, right? So it will be usually a enzyme, okay. So it is involved in the metabolism. So here they are given the example, that is a starch synthesis in a pea, okay, and phenylketonuria and the sickle cell anemia. What happened in the starch synthesis in the pea? Let us see. How many of you know that? So the starch synthesis in a pea is a pleiotropy, guys. See here. So starch synthesis in a pea, for example, it is having capital B and the capital B. Okay, guys. So it will give, it is having the more starch, right or not? So it is having more starch, more starch. That is a one character, right? And it is also responsible for shape of the pea, shape of the seed, right? It is also responsible for shape of seed. Now you tell me, guys, it is having the capital B, capital B. Now it is having the round shape or the wrinkle shape. Can you tell me whether it is having the both the allele, so that is the dominant allele, whether it will give the round or the wrinkle? Tell me, guys, in the chat box. Tell me in the chat box, do you understood or not? Round, right? Yes, guys. So whenever it is having the capital B, starch synthesis is more. That is the one condition, one characteristic. And another one is the shape of the seed is differ. So whenever the starch synthesis is more, so then it will give the round. Okay, then coming to the capital B, small b. Capital B, small b. Capital B, small b. Then shape of the seed is vary, right? Starch synthesis is the intermediate. So now you tell me in the small b, what will happen? So in the small b, what will happen? Very good, very good. So yes, Nandish, wrinkle seed will come, right? A wrinkle. We have studied about the green and the wrinkle, right? So it will give the wrinkle seed, and the starch synthesis also very less, right? So now both the character is maintained by the capital B and the small b gene, right or not? So that is a single gene, right? The single gene is having the two, yes, only three phenotypes. Yes, guys. So that's why we will call it as a pleiotropy. Pleiotropy. Okay, guys. Yes. So yes, multiple phenotype means that is only no. If it is only three phenotype also, yes, that is a multiple phenotype only. Okay, guys. So then coming to the phenylketonuria, let us study about that. Okay. And one more thing. So here there is a slide is there. So why Mendel works remains unrecognized? Can anyone tell me, guys, why the Mendel work was unrecognized? So when the Mendel was working on the time, there is no phone and the WhatsApp and the Facebooks or nothing is there. Very good, Lakan. Very good. Correct. So phone, WhatsApp, and Facebook is not there, right? So now we can, what are the experiment I have did? I can immediately send a message in a group. Now I have did this experiment like that, right? But in that case, there is no communication was not easy on that time. Newspaper and nothing is there, not much famous on that time, right? And coming to the, we know that he is having the mathematical approach, right? Mendel is having the mathematical approach, but all of the biologists are not having that much of the mathematical approach. They didn't understood what Mendel is telling on that time. Okay. So then the concept of the gene. So where it is not continuous, no, where here it is having a discrete form. We will call it as discontinuous, right? So that is the one reason he could not able to explain that to the another scientist. And the last one was he is not having the any proof for the existence of the factor. You know that he used to tell that some factor is there that is transmitting from the parent to the offerings, but he don't know that what is that factor. He can't physically take that factor and he can't show that factor to the other scientists, right? So that's the reason the Mendel work was unrecognized on that time. So that's why Mendel is always used to tell that my time will yet to come. My time will yet to come. Okay, but that time is came after he's died. Okay, after his death only the time has been came. That is in the 1900. In the 1900, so the three scientists independently recognized the Mendel's work. So that is De Vries, Correns, and the Testmark. In one of the NEET exam, they have asked this question, guys. They have asked this question. Don't ignore the scientist name. Okay. So De Vries, Correns, and Testmark independently recognized the Mendel's work. Okay. How many of you understood, guys? Independently, you know that. What do you mean by independently, right? Yes. So, for example, I am working independently in my laboratory, and Nandish working in his laboratory independently, and Lakan is also working independently in his laboratory. So then we come to know that some of the statements said by the Mendel was correct, right? So it was suiting to this experiment. So then we recognized its work independently. Right, guys? For Nandish also that thought came, and for Lakan also that thought was came, and for me also that thought was came. Right. So this is how they independently recognized the Mendel's work. Okay. Fine. So let us go to the chromosomal theory of the inheritance. Always remember, guys, here they may ask you the scientist name. Can anyone tell me what is the scientist name of the chromosomal inheritance? Yes, yes, very good. That is Sutton and Bowry. How come? Okay, so let us study about the chromosomal theory of the inheritance. So similarity between the Mendelian gene and the chromosome. So what they will tell in the chromosomal theory of the inheritance is, so there is a similarity between the Mendelian gene and the chromosome. Okay, guys. So Mendelian told that during the law of segregation, right, how the gamete will segregate during the gamete formation in the meiosis, right, guys. The same thing happen in the chromosome also. Both the homologous chromosome will segregate during the meiosis. The same thing he also told. Okay. So see here, the chromosomes are occurs in the pair. There also he told that genes are in the gene. The alleles are occur in the pair. Right or not, guys? The alleles are always occur in the pair. Yes, Sutton and the Boveri. Yes. So the alleles are occur in the pair. Right. So during the gamete formation, how the capital T and the capital will segregate. So here also what will happen? So the segregation of the chromosome will take place. In which stage the segregation of the chromosome will take place, guys? In the yesterday's class we have learned. In which phase? In which phase? I'm asking you which phase? Anaphase, right? In the anaphase, the chromosomal segregation is not happening. If the segregation was not happen, it will lead to the ploidy, right? It will lead to the ploidy. See here. So that is only he told that, right? So what happened? So when it is segregating, it is a haploid in number, right? So the haploid, so where the diploid will be restored after the fertilization, right, guys? So here the chromosome number will also restore after the fertilization. Okay. So that is what Mendel's told that. Okay. Sorry. That is what they have told that in the chromosomal theory of the inheritance. So similar, how the gamete will separate? So like that only chromosome will also separate in the homologous chromosome, guys. Okay. So chromosomal theory of the inheritance. So and it was where the proof, where the proof was given by, the theory was given by the Sutton and the Boveri, right, guys? Theory was given by the Sutton and Boveri, where it was proved by, who is the proved, guys? Can anyone tell me who proved the chromosomal theory of inheritance? Scientist name will come in the exam. Always remember, if they ask about the theoretical explanation, that is Sutton and Boveri. If they are asking about the experimentation, that is a T.H. Morgan. Okay. So yes, T.H. Morgan have did this experiment by taking the Drosophila. By taking the Drosophila, that we will call it as queen of genetics, or we'll call it symbol of genetics, right? Yes, fruit fly. Yes, fruit fly. Okay. So what happened here? He's taken the Drosophila for the experiment. Why he's taken that? Same thing, guys. It is having the very simple. It can grow in the very simple synthetic media and a short generation of the time, right? And the breed, it can breed. Whenever we are talking about the plant, the life cycle should be less. Okay. So whenever we are talking about the animals, it is having the it should be breed in the all the time. It should have breed in the all the time. So then we will take that as a experimental. Okay. Then whenever the one mating is happened, so it will give the hundred mates, sorry, hundreds of the progeny. Okay. So for that also we will take, and where male and the female flies are easily distinguishable. Okay. So where male is smaller than that of the female. Okay. And so the hereditary variation is present. Right, guys? In the last class also we have studied that in the sexual reproduction, hereditary variations are present, right? Yes. Yes. So there is a continuous variation is present. So you know what? You know, Nandesh. Continuous variation is not present in all the character. In all the character, that is not depend on the humans or the fruit fly. Okay, it is depend on some character. Some character. Continuous variation. Okay, in some particular character we can see the continuous breeder like that. Yes. You're talking about the breeder. No, yes, continuous breeder part. Yes, fruit fly are the continuous breeder. Okay. So let us talk about what you mean by linkage and what you mean by recombination. In the last class, we have discussed this, guys. Very good, Sahul, correct. So now you have to tell me, guys, if the two genes are here, you can see. Now you have to tell me. In the last class, we have discussed this. You also told me this. Now this A gene is here and B gene is here. Okay. Now you tell me whether recombination will happen or not. Now you tell me, guys, recombination chances is high or low? Now you tell me, just recombination chances is high or low? Tell me in the chat box. How come low? How come low? Recombination? How come the recombination chances are low? Because A and B will segregate independently, right? See here, guys, what happen always? So the linkage, where the linked genes always remember, guys, I have told you that linked genes will not show the law of independent assortment. Yes or no? Very good, Sahil. Very good. So when the genes are very far away, on the time recombination will be high. Okay, recombination will be very high. Okay. So those we will call it as the unlinked genes. The genes are not linked, right? So if you take Nandi, if you see here, I have told you in the last class that if the two genes are here, you can see here in the diagram only you can see it. Capital A and capital B genes are together, right? Whenever they are together, whenever they are going, they will go together only, right? They will go together only, right or no? Can it will follow the law of independent assortment, linked genes? Can you tell me, Nandish? Linked gene, can it show the law of independent assortment? Yes, Nandish. In the last class, we have discussed about the shopping and everything. No, that is only. It will not show law of independent assortment, right, guys? So then, in the linked gene, the chances of recombination is very less. Okay, chances of recombination is very less. In that case, what will happen? So the more progeny will be of parental type. Okay, guys, more progeny will be of parental type. Okay, so when we are talking about the genes which are far away, very low, very large distance is there means, so it will independent, it will assort independently, independent assortment will take place, and here you can see the recombination also. Here you can see the recombination will be very high in this case. Okay, so what do you mean by recombination type? So the generation of the non-parental gene combination, we will call it as the recombination. Okay. So recombination occurs due to the independent assortment or the crossing over. Right, guys? So whereas the recombination is not present in the linked gene. Right. So that's why it will not show the independent assortment. How many of you understood, guys? How many of you understood? Give me a thumbs up if you understood means. Hitesh, Nagveni, Nandish, Shanta, how many of you understood this pa? Give me a thumbs up if you understood this means. Lakand, Sahul, do you understood pa? Once again, okay. So just think here. So when the genes are linked means whenever they are together, whenever these genes are together, this will not, it will not assort independently. Means it will assort dependently, right? So this will not assort independently. It will go together only, right? Assort dependently, right, guys? Whenever they are very far away, I have told you in the last class, when me and my friends are very far away, right? I am in Bangalore, my friend is in Andhra Pradesh. Can we go for shopping together? Not at all possible, right? So Nandish told that, ma'am, you can do the online shopping. Except online shopping, we can't go together physically, right, guys? So that is the same thing happen here. When the two genes are together, they will go together only. They will not go independently, right? So that is why in the linked gene, what will happen? Recombination is very less. Recombination is very less, right? They will not do. They will not do any creative work, right? So because they are together only, right? They will do everything with the together only. So that is the reason. Hitesh, do you have any questions or do you understood, Pa? So Nagveni, nothing ma. Recombination means what happened, when the two chromosomes, no. See here in this diagram, when the two genes are very far away, okay, when the two genes are very far away, they will assort independently, right, ma'am? Whenever they assort independently, it will lead to the recombination. So non-parental type, it will give the non-parental type. Okay, so that we can see in the ours also, right, in the humans also. So where we are not like similar like a parents, right? So then coming to the genes which are together only, so the distance is very less. The distance between are very less. So they will assort independent, they will assort dependently only, they will go together only. So that is the reason recombination will not happen in the linked gene. And the linked gene will not show independent assortment. Wherever the recombination is happening, there the genes will show the independent assortment. That's it. Two things you have to remember here. That's it. Now you understood, Nagveni. Lakan, do you understood now? Lakan, Nagveni, do you understood, pa? Or you tell me once again, I will explain you, no problem. Lakan, do you understood? Lakan, Sahul, Lakan. Okay. So Nandesh understood. Lakan, if you didn't understood means tell me once again, I will explain. Okay, guys. So you know that how the recombination is happening, right? Now you tell me which phase of the cell cycle the recombination will happen. Fast. I need answer from you in the chat box. Lakan, tell me in which phase of the cell cycle the recombination will happen, in the meiosis or in the mitosis? Thank you, Sal. Okay. Lakan, you didn't understood or you understood, pa? Lakan, do you understood or not? If you didn't understood means tell me once again, I will tell you. Very good. In the meiosis, the recombination is going to be happen, right? In the patchetine, right, guys? The recombination is going to be formed in the patchetine state. In the prophase, that is in the patchetine, right? Yes, guys, in the patchetine. So now, yes, Lakan, in the anaphase. Yes. No, not in the anaphase. That is in the meiosis. In the meiosis, the recombination will happen. Which is the enzyme, guys? Which is the enzyme responsible for the recombination? Lakan, always remember. Not understood. Okay, I will tell you once again the recombination, guys. Okay, 1 minute. Where I can explain. Okay. See here, in this slide, I will explain you here. Lakan and everyone understood here. Okay. So if I take, these are the non-linked gene. Non-linked. Okay, guys, non-linked. Here I am taking the linked genes. Okay. So in the non-linked gene, what happening? So this is the one character. This is the one gene. It is responsible for the round. Okay, guys. This is the another gene. This is another gene which is responsible for the shape. Okay, guys. Both are very far away. Okay, guys. Both are very far away. So then coming to the linked gene. So here you can see the seed. So that is only same seed color you can take here. Okay. So it is having the one gene, so that is responsible for the green color. Okay, sorry, wrinkle shape. Okay, wrinkle. And there is another gene which is responsible for the color, green color. Okay, so now I have told you that in the non-linked gene, so here it will be the independent assortment will take place. Independent assortment, right, guys? So here what will happen? So no independent assortment. No independent assortment. If the independent assortment is taking place here means what you will get, guys? So you will get the round yellow, right, guys? Round yellow you will get, and you will get the wrinkle yellow, wrinkle yellow, right? Here you can see in the linked gene, what is the one type you are getting, guys? Only parental type you will get, right, guys? Only parental type. See here, linked gene, both the wrinkle shape and the green color will go together only. Okay. So that's why in all the progeny, we can see only wrinkle and the green. Wrinkle, this is a wrinkle and green, right? Because they are not going differently, right? They are not assorting independently. Both the thing will coming together only. Which are the wrinkle seeds are there? So that is having the green color only. That we will call it as a linked gene. Linked gene. How many of you understood now? Tell me, guys, how many of you now understood? Lakhan, do you understood now? Lakhan, do you understood now? Okay, that is what we will call as the linked gene. So let us go to the mutation. What is the mutation? All of you know that, right? The sudden change in the DNA, we will call it as a mutation. Right or no, guys? The sudden change in the DNA, we will call it as a mutation. Right. In the mutation, we can see for yours, so many mutations are there, guys. Okay, so many types are there. But for your syllabus, only two types of the mutation is there. One is a point mutation. Second one is a frame shift mutation. Frame shift mutation. Let us talk about the point mutation. Okay. So what do you mean by point? Point means where the mutation is happening, only change in the single base pair. Single base pair. Example is sickle cell anemia, where the single base pair change will lead to the mutation. For example, you can see here in the single base pair mutation, we are having the three types of the mutation, guys. Okay. One we will call it as a silent mutation, and another we will call it as the nonsense mutation, and other thing we will call it as a missense mutation. So just listen what I am telling. Okay. So what happened in the silent mutation? So here the TTC is there. Okay, in the normal DNA, TTC is there. So in place of TTC, in the place of C, I have added the T. Okay, guys, I have added the T. Okay. So now the amino acid before the mutation, it is giving the lysine only. Okay. After the mutation also, after the silence mutation also, it is giving the lysine only. Right, guys? Is that any amino acid changes has been taken place in the silent mutation? No, always remember. So in the silent mutation, what will happen? The mutation will lead to the same amino acid only. So where the nucleotide was changes, but it will give rise to the same amino acid only. Why? You know that the single amino acid was coded by more than one codon, right, guys? So that is the reason. Then coming to the nonsense mutation, what will happen here? Nonsense, right? What do you mean by here nonsense? See here, where there is a single base pair change leads to the stop codon. Okay, where only the mutation in single base pair will lead to the stop codon, where the synthesis is going to be stopped. For example, if you see before the mutation, that protein is giving, right? That protein is having like 300 amino acid, 300 amino acid. So when it is happen the point mutation, that is a nonsense mutation, that is having only like 150 amino acids. So what happened to the another 150? Where the mutation was happened in the 150th in the 150 amino acid, where the 150th amino acid was changed to the stop codon. Stop codon. So now what happened? The amino acid, the where the protein synthesis was stopped there only. Right, guys? Protein synthesis was stopped there only. Then it is given the stopping of that protein synthesis. Right? So always remember when the nonsense mutation was happened, where the mutation in the one nucleotide that will lead to the stop codon, where the protein synthesis was stopped there, where the the protein which is containing the amino acid number was very less in the nonsense mutation. So they may ask you the question like this, guys. Okay. So in the normal protein, it is giving the 300 amino acid. Okay, guys. So in the mutation, the mutation was happened and it was giving only the 150 amino acid. 150 amino acid. Now you tell me what type of the mutation it is. Tell me guys, I need answer from you. What type of the mutation it is? Tell me fast. I need answer from you. In the normal, it is giving 300 amino acid. So now mutation, it is giving the 150 amino acid. What type of the mutation it is? Sahul, Lakan, tell me in the chat box, pa. Nonsense, right? Nonsense. Mesense we have not yet studied, Nandish. Nonsense. Nonsense mutation. Where it is leads to the stop codon. Where the mutation will leads to the stop codon. That is a nonsense. Nonsense. Remember, nonsense. Very good, Lakan. Very good. Yeah, frame shift mutation also we will study. Okay. That is not a frame shift mutation. Okay. Frame shift mutation is different, pa. Where addition of the nucleotide or deletion of the nucleotide, where substitution of the nucleotide, that we will call it as the frame shift. We will come to that. Then coming to the missense mutation. So here what happening? So here, for example, before the mutation, it is coding for the arginine. Okay, guys. Before the mutation, it is coding for the arginine. Arginine is a basic amino acid, right or no? Arginine is a basic amino acid, right? So, yes, Nandish. Nandish, observe here what happening. So, in the DNA, what is happen? It is having the ATG, ATC, right? The mutation was happen where A is replaced to the U, okay? U. So, in the mRNA, we are getting UAG, right? So, here it should code something, right? But what happening here? It is leading to the stop codon. The mRNA has to synthesize, right? In between only stop codon is inserted here. UAG. UAG was, so this is the normal. So in the normal, UAG is at the last, right? So here some mutation was happened that will lead to the stop codon in the middle only. In the middle only. Okay. So due to that, what happening here? So it is giving the stop codon in the middle only, where protein synthesis is stopping in the middle only. Right. So that's why that protein is having very less of the amino acid compared to the normal protein. That is in case of the nonsense mutation. So now you understood, Nandish. So then coming to the missense mutation. Before the mutation, it is giving rise to the arginine, that is a basic amino acid. Okay. So after the mutation, what happening? It is giving, where here you can see the mutation was happened. In place of the G, C was inserted. So in place of the, here you can see, in place of the C, G was inserted. Before it is giving the arginine, before it is giving the lysine, now it is started to give the arginine. Okay, guys. So what happening in the missense mutation is the same, not the same amino acid will come. Here the same amino acid will not come, but same characteristic amino acid will come. For example, before the mutation, it is giving the glutamic acid. Okay. After the mutation, it is giving the aspartic acid. Okay, guys. If it is aspartic acid means both are acid only, right? Both are acid only. There will be a no much difference in that. So that we will call it as a missense mutation. Okay. So then coming to the another mutation, another I will give an example. So arginine. Arginine is there. Sorry, before the mutation, lysine is there. Okay, guys. So that is also basic amino acid. After the mutation, it will give rise to the arginine. Arginine also basic amino acid only, right? So there is no much variation in the protein structure. So that is what we will call it as a point mutation. That is a missense mutation. The amino acid will be same, but character is different. Okay, amino acid where arginine is there means it will give the lysine. Aspartic acid is there means it will give the glutamic acid. Yes, Nandish, I will tell you. So now let us come to the frame shift mutation. What happened in the frame shift mutation? Deletion or the insertion. So here you can see deletion or the insertion will give rise to the frame shift mutation. Okay. So here you can see guys, beast. Okay. In the beast is the normal one. Okay. So if I do substitute something, if I do substitute F. Okay. So here I am adding, here I am substituting the B with the F. Okay. The meaning was changed, right? So now it is giving the feast, right? The same thing happened, guys. For example, methionine. For example, AUG is the methionine, right? It will give the methionine. Okay. So for instead of the AUG, if I give UUG, okay. So it will give rise to the phenylalanine. phenylalanine, right or not, guys? The changing the frame, giving the different amino acid. So usually the frame shift mutation will give rise to the cancer. Observe here. What happening here? I have told you. AUG is there, right? So if I substitute A into the U, what will happen? UUG, right, guys? So now UUG means what will happen? Now it is coding the methionine. Now it is started to code the phenylalanine, right or not, guys? The amino acid was changed, right? The meaning was changed. The same thing happening in the frame shift mutation. Either it may be a substitution, or it may be a addition of the nucleotide, it may be a deletion of the nucleotide, or inversion, where it is giving the different amino acid, where it is changing the frame of the amino acids. Okay. So that we will call it as the frame shift mutation. Okay, guys. For example, if you see AUG is there, UAA is there. Okay. So now I am inserting the one amino acid here. So one nucleotide here. Now the frame was changed, right? Now it will be a AAU, GUA, right, guys? Before it is AUG and UUA, right? So now it is giving AAU and the GUA. Okay, guys. So that we will call it as the shifting in the frame. Okay. When the frame was shifted, it will give rise to the different amino acid. And what are the mutagens? Here the example is the 1 minute. Yes. Yeah, the example for the point mutation is the sickle cell anemia, right, guys? Sickle cell anemia, where the one nucleotide, where the glutamic acid was replaced by the valine. Glutamic acid, remember here, glutamic acid was replaced by the valine in the 6th position, right, guys? So that we will call it as a sickle cell anemia. In the coming slide, we will study about that in detail. So let us go to the mutagens. So mutagens, we know that mutagens are the agents which will cause the mutation, right? So there are the two types of the mutagens. Physical mutagens are there and the chemical mutagens are there. In the physical mutagens, we will see the ionizing radiation. Ionizing, right, guys? In the ionizing radiation, we can see the X-ray and the gamma rays, right, guys? X-ray and the gamma rays will come under the ionizing radiation. In the non-ionizing radiation, you will get the UV rays, right, guys? When coming to the chemical mutagen, tobacco, mustard gas, phenol, formalin, acetic acid, and ammonia, these all the things are the chemical mutagens. Okay, fine. So then coming to the karyotyping. So that is the sex determination. Okay, guys. Always remember that what do you mean by karyotype means the arrangement of the chromosome based on the in the decreasing order of their chromosomal size. Okay, the arrangement of the chromosome based on their decreasing size of the chromosomal size, we will call it as a karyotype. Okay, so let us study about the sex determination. All of you know that when you're talking about the human, so totally we are having the 46 chromosome, right, guys? 46 chromosome. In that 44 are the autosomes, and the X and Y we will call it as the X and Y are the sex chromosome. If it is a male is having the heterogametic, that is the X and Y, and the female is having the X and X, right? So always remember homomorphic. Homomorphic means X and X. And heteromorphic means it is having the two types of the gamete, that is the X and Y. Okay, guys. So let us study next. So male heterogametic. This is very important, guys. So listen here. Male heterogametic. The examples are male heterogametic. Male heterogametic means what happened here? The way male is producing the sex chromosome, we will call it as an allosome. Yes, guys. So what happened here? See here. When the male is heterogametic. Male is heterogametic means he will produce two types of the gametes, either X and Y or X and another zero. That's it. Okay. See here. Here you can see, and female will produce only one type of the gamete. So now you tell me, in the heterogametic male, who is the responsible for the baby sex? Who is the responsible for the offspring sex, guys? Male, right? Male is responsible, right? Yes. So observe here, the examples are humans. Humans are example for the heterogametic male, and homogametic female. Okay, and also the grasshopper and the cockroach. Grasshopper and the cockroach. And X and X and the X and Y type of the sex determination we can see in the humans. So where X and X will be the female, they will produce the homogametic, whereas the X and Y will be the male, they will produce the heterogametic. And it will be similar in the Drosophila also. Remember guys, Drosophila also will show the X and X and the X and Y sex determination. Then coming to the grasshopper, it will give the X and X will be the female, whereas the X and O will be the male, X and 0. Okay. So now what will happen in the grasshopper guys? So the grasshopper male is having one chromosome less than that of the female. For example, grasshopper is having the grasshopper male is having the 45 chromosome. Okay. 45 chromosome, that is a male. Okay guys, because only one X is there, right? Only one X is there. Coming to the female, female is having the 46 chromosome because 2X is there, right guys? So that is the reason. Okay. So grasshopper and the cockroach. So where the female is having the homogametic. So whereas the male is heterogametic, where it is having the one chromosome less than the female. And we can see the other type also, that is the female heterogametic. In the female heterogametic, what happening here? So the example is the bird. Example is the bird, guys. So here you can see the ZZ, that is the male. ZZ will be the male, whereas the ZW, that is the heterogametic female. Heterogametic female. Can you tell me now, who is the responsible for the sex in the bird? Who is responsible for the sex determination in the bird? Either the father or mother, guys. Tell me. Can you tell me? Hen, right? Female. Female is responsible for the sex in the bird. Okay, guys, always remember that. Females are heterogametic. They will produce the Z and W type of the gamete, whereas the male will produce only the ZZ type of the gamete. Okay, guys, fine. So there is another type of the things is there. So that is a haploid diploid. Haploid diploid, that we can see in the haploid diploid sex determination. We can see in the where, guys? In the bee, right? In the bee we can see the haploid diploid. What happened here? The bee. So in the bee we can see the worker and the queen, right? Worker and the queen are produced by the fertilization. They are diploid in nature, 2n in number. Okay. So when we are talking about the drone is a male. So male is formed by the mitosis, only mitosis. There is no fertilization will be happen. So they are haploid. Males are the haploid. Always remember, guys, males are haploid. Okay. Yes, drones are the haploid, whereas the female, that is the workers and the queen, those are the diploid, 2n, right? So they are come from the fertilization. Okay, guys. So and one more thing you have to remember here. So that is the female will undergo the meiosis for producing the gamete, whereas the male will undergo the mitosis. So males will undergo the mitosis to produce the drone. So yes, so that's why in the male what will happen? So there is no father and there is no son. Okay, there is a grandson is present, whereas the grandfather is present. Okay, guys, fine. So these all the things we have studied. So and thinking how we discovered the X chromosome and haploid diploid sex determination we have studied. So always remember, guys. So drone is having the 16 chromosome. Okay, guys. Drone is having the in the diploid honeybee, it is having 32. Okay, 32 chromosome. So when it is a haploid, the haploid is having the 16. Means always remember the drone is having the 16 chromosome, whereas the worker and the queen is having the 32 chromosome. Okay, guys, 32 chromosome. Yes, Lakan, I will complete in 5 minutes. Okay, guys. So then coming to the genetic disorder. So there are the Mendelian disorders are there and the non-Mendelian disorders are there, right? So if you take the example, hemophilia, color blindness, beta thalassemia, sickle cell anemia, phenylketonuria, and cystic fibrosis. These all are the Mendelian disorder, guys. Okay, Mendelian disorder. So these are comes due to the mutation of the DNA. Always remember, mutation of the DNA. Okay. So when you are talking about the chromosome number changes, so that is due to the disorder. So that is a chromosomal disorder. That is due to the ploidy. Ploidy. Okay. So one is the Down syndrome, Turner syndrome, and the Klinefelter syndrome. Okay, guys. So these are the genetic disorders. We can complete. Ma'am, what do you mean by Turkey? Turkey. Yeah, white color pa. Turkey means white. They are the Turkians are white. We will call it as the albino, right? For the albino, we will call it as a turkey. Turkey pa. That's it. Turkey sex determination. Okay, I will let us discuss that in the tomorrow's class. I will check and I will tell you in the tomorrow's class pa. Okay, Nandesh? Fine, guys. Read well for the exam. Take care. All the best, guys. Take care. Bye bye. Hi, Nandesh.